Hi, my name is Jessica Mosmann, and I'm going to present our work called Genetic Characterization of HPV16 E6 and LCR variation of oral lesion from Argentinian patients. We know that HPV is the most frequent sexually transmitted infection worldwide, and new lines of study show that HPV may be an independent risk factor for several subtypes of head and neck cancers. Most studies show that some point mutations on HPV16 genome have a higher potential to generate precancerous lesions if they are in the viral regulatory long control region, LCR, and E6 gene. So our objective was to describe the genetic variability of E6 and LCR region in the HPV16 sequences from oral cavity lesion samples. We use oral swabs from benign lesion, potentially malignant oral disorders, and oral squamous cell carcinoma. These samples were taken by dentists, and the lesions were identified previously by pathology analysis. We did DNA extraction, and then we analyzed the samples using specific PCR to E6 and LCR HPV16 genes and Sanger sequencing. Genetic variations were analyzed using MEGA11 software. These samples were involved in the previous project in which we studied 318 oral swabs. So we use only HPV16 genotypes to the present study. And we could be obtained 23 oral sequences for E6 gene and 8 sequences for LCR gene. Okay, the more frequent point mutations to E6 was T350G in 18 sequences. And some studies have reported that this variation is associated with an increased risk of development persistent infection that would stimulate cancer progression. Other point mutations were identified in these genes, and E6 is a coding region, so some point mutations could produce an amino acid changes. Here we can see that all point mutation detected have produced an amino acid changes. On the other hand, when we analyzed statistical correlation, we did not find significance between the mutation and lesion types, patient sex, and tobacco and alcohol habits. For LCR gene, the more frequent point mutation detected was G7521A in 7 sequences. In the LCR region, this position is a binding site of the transcription factor YY1. This is a regulator of transcription of viral oncogenes. So the changes in this site could result in high levels of HPV oncogenes. Remember that LCR is a non-coded region, so here we did not see amino acid changes. Here we did not find significance between mutations and patient sex, lesion types, and tobacco and alcohol habits. As a conclusion, this is the first preliminary study of HPV16 mutations obtained from oral lesion from Argentinian patients. And point mutation associated to malignancies were identified in benign lesions, potentially malignant oral disorders, and oral squamous cell carcinoma, highlight the importance to know the lesion to offer a correct treatment to the patient. HPV16 infection is a potential risk factor to oral lesions, and we think the knowledge of genetic variations could be an important point to oral cancer prevention. And that's all. Thank you.
I am presenting the study and integrated analysis of genomics repositories data identified AH receptor germline pathogenic variants in patients with head and neck cancer. This study was performed by researchers at the Universidad Cooperativa de Colombia and Universidad Industrial de Santander. Head and neck cancer is the sixth most common neoplasia according to Globocam, and is one of the most common causes of death globally. In 2020, more than 930,000 new cases and 460,000 deaths caused by head and neck cancer was reported. In Latin Americans, more than 25,000 people died of head and neck cancer. In Colombia, 3,148 new cases was reported and 1,343 people died of head and neck cancer. The aryl hydrocarbon receptor, AHR, is a transcription factor encoded by the AHR gene. AHR induces the activation of xenobiotic metabolizing enzymes, and it has been associated with the regulation of multiple signal transduction pathways. Polymorphic variation in AHR could affect the inducibility of target genes encoding regulatory mechanisms and contribute to cancer susceptibility. The objective of this study was to compile genetic variants in the AHR gene reported in cancer genomic repositories, with a focus on head and neck cancer and the Latin American population. The methods. A search for variants in AHR was performed in 5 repositories, focusing in head and neck cancer and Latin Americans. The databases included were COSMIC, ICGC, TCGA, NOMACT, and the GWAS catalog. The data was consolidated in Excel, followed by pathogenicity analysis in silico of the variants, and classification by primary site, types of variants, and population. Variant pathogenicity was evaluated using FATOM, BEP, SIFT, and Polyphen. As a result, we consolidated the data from COSMIC, ICGC, TCGA, NOMAD, and the GWAS catalog. A total of 720 variants in AHR were found among the 5 repositories. 1,301 variant was reported in COSMIC. 1,124 variants were reported in ICGC. 230 variants were reported in TCGA. 4,359 in NOMAC, and 6 variants were reported in the GWAS catalog. After removing duplicates, 280 variants remained for the following analysis. Variants in AHR were reported in 25 primary sites, mainly in breast, skin, and liver. AHR variants were reported in head and neck cancer, but only 17 variants were reported in COSMIC, 8 variants in ICGC, and 7 variants in TCGA. From the total of 280 variants identified, 63% were intronic variants, 13% were missense variants, and 5% were located in the 3' UTR. From these, 19 variants were reported in head and neck cancer patients. 8 variants were missense, 8 were intronic, 1 frame shift variant was identified, 1 in frame insertion, and 3 UTR. Through the in silico analysis, 10 of the 19 variants reported in head and neck cancer patients have moderate or high impact and which could influence the function of the protein. 2 of these variants are insertions and 8 are single nucleotide polymorphisms. Among the total 280 variants, more variants in AHR were identified in individuals from United States, followed by individuals from Australia with 21% and 15% of frequency, respectively. According to the ethnicity, which is reported by TCGA, most of the variants were reported in white individuals, and 14% of the variants were and the ethnicity was not reported for 14% of the variants. Talking about head and neck cancer specifically, according to ICGC, 75% of the variants were reported in individuals from United States and 25% in individuals from China. According to TCGA, the variants reported in head and neck cancer belong 86% for white, non-Hispanic or Latino, and 14% were identified in African Americans. We wanted to know what was happening with the Latin American population. So we identified the HRR variants reported in this population. 39 variants were reported in Latin Americans. From these, 11 variants were classified with high or moderate impact on the protein function. Conclusions. 19 variants were reported in AHR for individuals with head and neck cancer. From this number, 10 may have an impact on the protein function according to the in silico analysis. ICGC, TCGA, and COSMIC repositories have informed variants in AHR in 25 primary sites. However, studies reporting AHR variations in the Latin American population are limited to 11 cancer sites: skin, uterus, colorectal, kidney, esophagus, prostate, stomach, liver, bladder, cervix, and ovary. However, none of these variants were found in Latin American individuals with head and neck cancer. The small number of variants in AHR that we identify in Latin Americans, 39 out of 280, might be related with the underrepresentation of this population in the genomics repositories. This indicates a necessity to include more Latin American individuals in genomics studies. A perspective of this study is to determine the effect of the variants identified as pathogenic by using cell and molecular biology approaches, and to evaluate the risk that these variants may confer in a future association study. We want to acknowledge Headspace Head and Neck Cancer scientific organizers and the financial support of CONADI, Universidad Cooperativa de Colombia, and Universidad Industrial de Santander. These are the references. Thank you.